Hello dear students, Karnataka PU board released 5 model papers for second PUC, and from biology subject here is the first model paper. I have solved all the MCQs correct answers I have marked. You can check all the 15 MCQs right answer has been marked. And remember, the 13th MCQ, the organism that reproduce only once in a lifetime, the right answer is salmon fish. And when you take plants, it is bamboo. Filling the blanks also, right answer written. Now in part B, 21 question is a diagram of two-celled pollen grain that everyone knows it. 22 4 assisted reproductive techniques I have mentioned: IVF, ZIFT, GIFT, ICSI and AI. And 23rd question, recombinants obtained in both Mendelian and Morgan experiment, yet the processes involved are different. Comment. So as you know, Mendel recombination is because of independent assortment of genes, whereas Morgan recombinant is due to the crossing over of homologous chromosome, that you need to write. And 24th question, name the methodologies used in Human Genome Project. It is ESTs, experience sequence tags and sequence annotation. And 25th question, name the evolution of any two plant forms through geological periods for tracheophyte ancestor. It is fern, then conifers, means gymnosperm, then angiosperm. 26th question, briefly explain the primary sewage treatment. Primary sewage treatment includes filtration and sedimentation. And 27th question, difference between ex situ and in situ conservation is very easy, everyone knows it. And for your kind information, second PC biology, all the chapters simple notes I have already uploaded in my YouTube channel. You can go and check it, free PDF also available. 28th question, diagram of monocot embryo. Then 29th, menstrual cycle definition. And the pituitary gland secretes hormone LH and FSH, and ovarian hormone are estrogen and progesterone. Then 30th question, write any three principles to be followed by the people to free from sexually transmitted infection. That is very easy. We need to avoid sexual contact with unknown person, sexual contact with multiple person, and using condoms before having intercourse. And if you have any doubt, we need to consult the doctor. That three reasons, the three principles you need to write. 31st question, list the three connotation of theory of special creation. So it says that special creation, theory of special creation says that all living organisms were created as they are today, and diversity is same as before, and earth evolved around 4000 years old. That's what theory of special creation says. 32 question, name the diseases caused by Wucheria elephantiasis, microsporum ringworm and plasmodium malaria. 33 question, mention the use of the following bioactive agent. Streptokinase is a clot buster used to treat heart diseases like myocardial infarction. Cyclosporin A is a immunosuppressive agent used during organ transplant, and statin is used to reduce the blood cholesterol level. 34th question, describe the steps of decomposition. Decomposition includes 5 steps: fragmentation, leaching, catabolism, humification and mineralization. That's what you need to explain. 35th question, male reproductive system diagram with labeling. 36th question, schematically represent two gene inheritance, means dihybrid cross you need to explain. Then 37th question, DNA is a genetic material in both prokaryotes and eukaryotes, yet the packing process in their cells is not same. Explain. As you know, prokaryotes does not have well-defined nucleus, whereas eukaryotes has a well-defined nucleus. So with the help of histone protein, how the DNA is going to pack regarding euchromatin, heterochromatin, you need to explain about nucleosome in 37th question. 30th question is very easy, difference between innate and acquired immunity. Then they mention the cells involved in accomplishing the acquired immunity. And there is a diagram. In this diagram, the name the molecule trapped in the lymph node is antigen at binding site A and B, and H2L2 means two heavy chains and two light chains of antibody. 39th question, process of recombinant DNA technology you need to write. Then the plasmid diagram has 39th A, explain the process of preparation of competent host for transformation with recombinant DNA. You need to explain heat shock method. 39b plasmid diagram is given. Here A is restriction enzymes present at ampicillin resistant gene is PST1 and a tetracycline resistant gene BAMH1. And how many cloning sites are present in PBR322? There are 8 cloning sites you can find that you need to mention. 40th question, genetically modified plants have been developed through core techniques of biotechnology. How are these genetically modified plants useful to humans? So you need to mention about they tolerate abiotic stresses, post resistance, the pest resistance, then post harvesting loss can be reduced, nutritional enhancement and mineral usage is enhanced. That points you need to mention. And 41st question, write an equation to calculate the population density at time t. I have given the formula nt plus 1 is equal to nt plus b plus 1 minus d plus e. And we need to explain. B means birth rate, I means immigration, D means death rate, E means immigration, N means population density. That's you need to explain. 42 question, correct answers I have labeled. You can refer the table. 43 question also I have marked the right answer for all A to D. And 44th question, DNA polymerase cannot transcribe a gene by itself because DNA polymerase needs sigma factor during initiation and rho factor during termination. That you need to explain. And transcription and translation are separate by site because transcription takes place in the nucleus and translation takes place in the cytoplasm, and both are not coupled because RNA processing is required, like splicing, capping and tailing for hnRNA. And which RNA polymerase enzyme involved in the transcription of 5S rRNA, that is RNA polymerase 3. If you need a PDF of this model paper, you need please comment me, then I will provide the PDF notes for you. Thank you.